Hello, welcome to this new lecture series on crop anatomy. In this lecture series, our lecture 11, and we will be looking at the stem. The stem of a plant is that part that we see outside of a plant. It enables a plant to carry its leaf, it enables a plant to carry its fruit, it enables a plant to carry its reproductive parts. Okay? Without the plant will be more like an underground thing. Okay, the stem enables a plant to be outside to receive sunlight. Is that aerial portion of a plant. The plant, remember we told you in the first lecture series that the plant divided by two parts majorly. We have the above ground part and we have the below ground part, and the stem is that part that is above ground that enables us to see what the plant leaf looks like, what the fruits look like, what the flowers look like. Okay, the stem is a part of the plant that grows ascending and in its area in nature, and also bears leaves and reproductive structures. The stem, together with the leaves which it bears, constitutes the shoots. It bears conspicuous node and internode and differ from roots in their vascular structure. This difference lies in the arrangement of xylem and phloem. The stem first originates from the stem meristem during the embryo development. Inside the embryo lies the stem meristem. We have the root meristem, the stem meristem, okay? And the stem arises from there during that point when the embryo is emerging, and then you're more likely when you're trying to say there's a germination taking place in the seed, and then you see the seed, the plant coming out, the new plant coming out of it. And say that fully developed embryo commonly consists of an axis. That is an axis, the hypocotyl root axis. So this axis bears at its upper end a pair of cotyledons and the shoot primordia. So as it comes out, you have those two cotyledons in the dicot, or one cotyledon for a monocot, and then with a shoot primordia. The seed plants are divided into two groups. So we have the gymnosperms and the angiosperms. Remember I told you gymnosperms are plants that have naked seeds, while angiosperms are plants that have seeds that have some covering on them. So but angiosperms are the focus of this crop anatomy lectures, because we are talking about crop. Those plants that are deliberately cultivated for man's consumption. So angiosperm is further divided into dicotyledons and monocotyledons. Now let's look at the anatomy of dicotyledonous stems. The epidermis. The epidermis consists of a single layer and is the outermost layer of the stem. It contains stomata and produces various types of trichomes. The outer cell wall are thickened and heavily cutinized and lacks intercellular spaces. It serves mainly for restricting the rate of transpiration and for protecting the underlying tissues from mechanical injury and from disease-producing organisms. Immediately after the epidermis lies the band of collenchyma. These cells are modified parenchymal cells with cellulose, which was thickened at the angles where three or more cells are in contact. The function is to serve as strengthening material in succulent organs which do not develop much woody tissues. Remember I've said this before, collenchyma comes before the formation of parenchyma and sclerenchyma. So however, collenchyma's job is to provide that structure, that strength to those new growing shoots, those new growing seedlings. Okay, it gives it that strength for the leaves to be able to stand erect. It's the ability of the collenchyma that gives that leaves, that the presence of the collenchyma that gives that leaves the ability to remain erect, the stem or the shoots to remain erect at that succulent stage. Okay, so it's strengthening material in succulent organs which do not develop much woody tissues, and in soft young parts of woody plants before the development of stronger tissues. These cells in stems sometimes possess chloroplasts and sometimes carry out photosynthesis. The cortex. The region that lies next to the epidermis, that means after the collenchyma band, is the cortex. The innermost layer of the cortex is the endodermis, also known as the starch sheet. Okay, we'll talk about more about the endodermis later. Okay, so the cortex consists of a single layer of cells which surrounds the stele and contains numerous starch grains. The parenchyma is found within the cortex. Their cells are living and contain moderate amount of protoplasm. When exposed to light, they develop chloroplasts and become known as chlorenchyma cells. Parenchyma cells of cortex in stems are near enough to the light so that some or all of them develop chloroplast and perform photosynthesis. Turgid parenchyma cells frequently help to give rigidity to the plant. They serve for slow conduction of water and food. It is a special storage tissue of plants. Sclerenchyma. The sclerenchyma cells are found in the cortex of stems. There are two varieties of these sclerenchyma cells. We have the stone cells and the sclerenchyma fibers. The sclerenchyma fibers are long, thick-walled dead cells and serve as strengthening material. I've talked about this before. We're just doing a bit of a recap. Stone cells give stiffness to the cortex. Now we're talking about the endodermis. It said it's an inner layer of the cortex and is consisting of barrel-shaped, elongated, compact cells, having no intercellular spaces among them. They contain starch grains, so they are termed starch sheets. Stele. This is the part of the stem inside of the cortex. It consists of three general regions: the pericycle, the vascular bundle region, and the pith. Combining those areas, we have the stele. The pericycle, this is the region between vascular bundles and the cortex. It is generally composed of sclerenchyma and parenchyma cells. Vascular bundles are seen in broken ring form. Each vascular bundle consists of three parts: xylem, phloem, and cambial layer. The xylem, which is formed before the activity of the cambium has begun, produce xylem and phloem cells, is called primary cells. That's xylem that comes first before xylem and phloem are produced. Phloem, the primary phloem of the dicot stems, consists of three types of cells: the sieve tubes, companion cells, and phloem parenchyma. There lies a layer of meristematic cells between the xylem and the phloem. It's known as cambium. It consists of a single layer of cells which by division gives rise to xylem cells toward the center of the stem and phloem cells toward the periphery. Pith rays. The vascular bundles are separated from each other by radial rows of parenchyma cells known as pith rays. They serve primarily for the conduction of food and water radially in the stem and for food storage. In a dicot plant, the center of the stem is composed of thin-walled parenchyma cells and is known as the pith. The cells have distinct intercellular spaces. Okay, so here we have the diagram of the parenchyma, the dicot stem. We have the transverse section and the longitudinal section. Okay, so this is not too clear, but you have to pay attention to each of these labelings. Okay, you see where the pericycle is, the pericycle, the vascular bundle, and the pith all make up to form what we call the stele. Okay, here is the pith. The pith is right within this place. So we have the epidermis. We have the epidermis made with comprising having the cuticle. We have the medullary, the space in between each vascular bundle. We have the cortex immediately after the collenchyma band, which is this. Okay, we call the collenchyma band hypodermis. Okay, so we have the pericycle, the oil ducts, and then if you come through this direction, you can see more information as to the pith. Like I told you, this is these are the vascular bundles, and this is a pith immediately after the vascular bundles. So this is how the dicot stem looks like. Okay, so take note of this information. They are very important for your understanding of crop anatomy. Let's look at the anatomy of the monocotyledonous stems. Apparently, the monocot stems are similar to dicot stems in having an epidermis, a cortex, and a stele. However, the cortex may be well developed and sharply marked off from the stele, or it may be quite narrow and inconspicuous. It is in the structure and arrangement that monocot stems differ markedly from dicot stems. Okay, so everything that the dicot has, all that we talked about, the epidermis, the dicot, the cortex, the sclerenchyma, parenchyma, and so on, they also have those similarities in terms of the monocot, but they differ in a few areas. And one of such areas is that the monocot doesn't have ring arrangement with respect to the vascular bundle. They are not ringed arranged, they are scattered. And also the stele is not, which comprises of all those things we talk about, is not differentiated into those distinct parts like the vascular bundle area, the pith rays. Okay, so the pith and the pith rays are not well defined. Okay, so sometimes the center of the stele is free from vascular bundles and is occupied by parenchyma cells, which as the plant grows up dries up and disappears at an early stage, resulting in hollow stem as seen in most grasses. Okay, if you have checked a grass, if you haven't checked one along your environment, you can literally check the stem of the grass and you might find it compressible with the inside being hollow. Okay, that means that the parenchyma cells have dried up. The vascular bundle of monocot stems are like those of dicot stem in consisting of xylem towards the stele and phloem towards the periphery. However, the vascular bundles of monostem do not possess cambium like that of the dicot, so hence, which means they will not exhibit any secondary growth. Okay, so each bundle remains more or less completely surrounded by a sheet of sclerenchyma cells, the bundle sheet. The phloem is made up of mostly sieve tubes and companion cells, and the xylem of vessels and wood parenchyma. So we have some distinct features we need to look at when we talk about the monocot stem from the dicot stems. The vascular bundles are many, much more than that of the dicot. Remember we talked about the diarch, the triarch, the tetrarch, the polyarch, the pentaarch and the hexaarch. Okay, but for that of the monocot, we have them from 6, 7 and above. Okay, the minimum is 6 and then above. Why that of the dicot is from 1, from 2 down to 5 or 6 thereabout. So they are numerous. Monocots have numerous vascular bundles, and stele is broken up into bundles, and vascular bundles are lines scattered in the ground tissue axis. For that of the dicot, they are arranged in a ring form. The endodermis is not found. There is no endodermis in the monocot. Why that the dicot we have? The cortex, pericycle, and pith are not differentiated because of the presence of scattered bundles throughout the axis. Vascular bundles are collateral and closed. That means they move in parallel direction. There is nothing separating them. Okay. After the xylem is the phloem, and they are closed with that bundle sheet we're talking about. They lack secondary growth. Leaf trace bundles are numerous. Vascular bundles are commonly oval in shape. The phloem is represented by sieve tubes and companion cells only. Phloem parenchyma is absent. Another distinct feature is that of the phloem does not contain phloem parenchyma cells in the monocot stems. The pith is not marked out, but it is present. The sclerenchyma hypodermis is present, and epidermal hair is absent. So this is an image of it. We have the epidermis, the ground tissue, the vascular bundles. We do not have epidermal hairs. Okay, let's look at it in the longitudinal section. We have the bundle sheet, the phloem, the xylem. We have the ground tissue, the vascular bundles. You see, they are scattered. They are not in ring shape. Why these areas comprise of the pith and the pith rays, not distinct division. So that is all for this lecture series, and I want to urge you to check out for the next lecture series, as we'll be discussing the anatomy of the leaf and the petiole. Thank you for this time. See you in next session.